Good evening, everyone. So, in this particular session, we are going to focus on looking at what we call hemopoiesis. So, hemopoiesis basically is just a process by which the formed elements of blood are going to be produced and they are going to be developing. So, this whole process that we call hemopoiesis involves the formation of the formed elements. So previously we said that formed elements of the body of the blood, we have got red blood cell, white blood cell, platelets, and so on and so forth. So this whole process of hemopoiesis just highlights on how these formed elements are actually formed, and they get to find themselves in the blood functioning as mature formed elements. So hemopoiesis involves three types of processes. So each formed element, like the red blood cell, white blood cell, as well as the platelets that are referred to as thrombocytes, they have got their own mechanisms in which they are produced. So first of all, when you're talking about red blood cells, also known as erythrocytes, their process of formation or their hemopoietic process is known as erythropoiesis. Erythropoiesis. Then when you're talking about white blood cells, also referred to as leukocytes, their process is known as leukopoiesis. Then finally, when you're referring to platelets, also known as thrombocytes, their process is known as thrombopoiesis. So in this particular video specifically, we are just going to be focusing on red blood cells, meaning we are going to just talk about the whole process of erythropoiesis. So we know that when the mother gets to be pregnant, then the fetus starts developing into the body. At some certain period of the pregnancy, there has to be this process, which is hemopoiesis process, has to start taking place, in which the formed elements of the blood has to start being formed so that they can support the growth of the whole body, the nutritional exchange, and also the supply of oxygen, and also the excretion of metabolic wastes, as we talked about the functions of blood. So you're going to find that by the time the pregnancy is reaching the 20th week, you're going to find that the process of hemopoiesis has to start taking place into the fetus. And there are actually sites in that fetus where the all processes of hemopoiesis tends to take place, because we know that that fetus is actually still developing from the fetus, going just like that. So it is still developing. So you're going to find that the sites of hemopoiesis tends to be shifting depending on the developmental stage. And as the fetus gets to now develop fully, and it is now time to give birth, you're going to find that now the child starts growing into a young adult or into an adult or reaching adulthood. You're going to find that the whole process of hemopoiesis is going to be shifting. So before we get to go into this process of hemopoiesis, specifically erythropoiesis, so that we can find out how the red blood cells are formed, there are actually sites, that the same sites that I talked about. Let's try to highlight them and see during what period do they come into play in terms of the formation of these formed elements of the blood. So we are going to have the first one. So first of all, we have got what we call intraembryonic structures as well as extraembryonic structures. So these structures are structures that are involved in the formation of these formed elements, are structures that carries out the whole process of hemopoiesis. So intraembryonic structures, these are structures or organs that are found inside the embryo that carries out the whole process of hemopoiesis. And extraembryonic structures are structures outside the embryo that carries out the whole process of hemopoiesis, in which these formed elements are actually formed. So we're going to find that the yolk sac is going to be the first structure that will be involved in the formation of these red blood cells. Leukopoiesis will be taking place, formation of white blood cells, thrombocytes, and so on and so forth. So we're going to find it is going to be the first organ that produces these formed elements, or red blood cells specifically. Then you're going to find that from there the spleen as well as the liver is going to take on. So you're going to find that within the first trimester, between the first trimester till birth, the yolk sac will be involved. Then after the embryo starts now growing, the fetus coming in, these structures will start coming in. Spleen and the liver will start now taking over the whole process of forming these red blood cells. Then from there, when the child is now born, the bone marrow will come into play. So the bone marrow will come in and take over the whole process of hematopoiesis, and it will become dominant. So it will become the site for the production of these formed elements. So you're going to find that in children, the marrow, which is involved in the formation of these formed elements, it is found in the appendicular skeleton, as well as the exoskeleton. So you're going to find that these skeletons, so the appendicular skeleton and the exoskeleton, these are just skeletons that are involving your limbs, your upper limbs and the lower limbs, the skeleton of the upper limbs and the lower limbs. Then the exoskeleton, it is the body trunk, where we're going to find the ribs, the thoracic cavity there, and so on and so forth. So that's where that's the skeleton of these two classes, so the appendicular as well as the axis. So I'm going to find that the bone, in fact, the whole lot of the bones are involved in the whole process of hematopoiesis. They are involved in the formation of these formed elements, the red blood cells, white blood cells, and so on and so forth. So they are going to be actively involved. Then you're going to find that as this child tends to grow, reaches an adulthood level, becomes an adult past 20 years, you're going to find that the whole process of hematopoiesis is going to shift. So you're going to find that certain structures that used to produce these formed elements are going to stop. For example, some of the structures of the appendicular skeleton, you're going to find that those bones of the appendicular skeletons, like the femur bones, the humerus bones, and also these other bones that you're going to find there, they are going to stop forming these formed elements. So you're going to find that only the proximal parts of these bones of the appendicular skeleton are the ones that are going to be involved in the formation of these formed elements. As someone gets to grow and reaches an adulthood level, that is somewhere past 20, 21 years, then this whole process of hematopoiesis tends to shift. So as we talked about, we have got intraembryonic structures that are involved in the formation of these formed elements, as well as extraembryonic structures. So these intraembryonic structures involves the spleen, the liver, as well as the bone marrow. Then extraembryonic structures, it is the yolk sac, because we know that the yolk sac is the one that more like houses the embryo as it is developing. So during the period when this embryo or these intraembryonic structures haven't yet grown or developed to carry out the whole process of hematopoiesis, the yolk sac carries out that process. So you're going to find that the yolk sac is going to be the extraembryonic structure, and the liver, the spleen, and the bone marrow are the intraembryonic structures. So the yolk sac is going to be the first site for the formation of blood. So now, after understanding this, we would like now to get into the whole process of erythropoiesis. So we're going to look at the erythropoiesis. We also look at what are the factors that tend to regulate the whole process of erythropoiesis, and also how those factors tend to play a role, and at what stages of erythropoiesis do those factors tend to carry out their functions. So before we get deep into the erythropoiesis process, we just have to understand certain terms. So we have got certain types of cells that are known as the stem cells. Then you also have another type of cells that are known as hematopoietic stem cells. Then you also have another type of cells that are known as committed progenitor cells. So these cells are actually very, very important, and you need to understand them because they will be more like the ancestry cells that are found in that bone marrow that are facilitating the formation of specifically the red blood cells, as we are going to dig into the process of erythropoiesis. So these cells are actually cells that are capable. So these cells, they are capable of differentiating into a specialized cell under specific conditions. So you're going to find that this type of cells, they are just there waiting. They are more like the way water is. You know that water is colorless. So you're going to find that any type of color that you put in, that's how water will become. So these stem cells, that's how they are. they are undifferentiated and they are capable of renewing themselves, and they are also capable of differentiating into a specialized type of cell under specific conditions. So we have got specific conditions. So this specific condition, it is the presence of certain inducing factors. So we have got inducing growth factors such as the interleukin 1, the interleukin 3, the interleukin 6, and also other ones. So these factors, they tend to give directions to these stem cells into the specific type of cells that they should differentiate into, so that they can form the required type of cell that the body is requesting for. So these stem cells are just capable of renewing themselves and also differentiating into specialized type of cells under specific conditions. So the specific condition are these inducing factors and other factors. We are going to talk about them. Then we also have these type of cells that are known as hematopoietic stem cells. So these hematopoietic stem cells, they are found in the bone marrow. So the bone marrow basically is of two types. We have got the red marrow and also the yellow marrow. The red marrow, it is the one that is actively involved in the formation of the red blood cells. And then the yellow marrow, it is the type of the bone marrow that was once, performing the whole process of hematopoiesis, but at some point they had to stop. That's why I find that these bones, like the bones of the appendicular skeleton, except the proximal parts of them, they tend to stop producing these formed elements. So they turn into the yellow marrow. So these hematopoietic stem cells, these cells, they will start giving rise to blood. They will give rise to all blood lineage. So meaning that these cells, they are the one now that are going to start differentiating. So when they differentiate, they'll form specific type of cells that will give direction. So when they form these type of cells, it will now be known to say, oh, here it is the red blood cell that is going to be formed. Here it is a white blood cell. Or here, these are actually what we call platelets that are going to be formed. So these hematopoietic stem cells, they are found in the bone marrow and they give rise to a blood cell lineage. So meaning they are going to give the formation of a specific type of cell that will be just in the lineage of the formation of red blood cells or white blood cells or platelets. So that's how they are. So they also function under specific stimulants, other inducing factors, and also conditions like these that we talked about there. But we're going to look at them in detail. There is a table that is showing all those. Then we also have what we call the committed progenitor cells, just from the term committed. They are committed in these relationships, these cells. So these cells, basically, they are restricted, or they are just restricted to a specific lineage, or they can only produce one specific type of blood cell. So you're going to find that we have got a certain type of committed progenitor cell that is called the CFUE, or the colon-forming unity erythroid. This colon-forming unity erythroid, it is from the term erythrocyte. So this erythroid, colon-forming erythroid, it is a type of committed progenitor cell that is specifically just involved in the formation of red blood cell. So meaning that this type of committed progenitor cell specifically just going to lead to the formation of red blood cell. So they're just going to be committed in that relationship that no matter what, I just have to form red blood cells. That's actually cool. Okay, so we now get to dig deep into the whole process of erythropoiesis without wasting much of the time. So erythropoiesis, as we talked about, is actually a process by which red blood cells are formed from the hematopoietic stem cell. And we know that these hematopoietic stem cells, they are found in the bone marrow, as we talked about, and they dictate which type of cells they need to form so that it can point to the lineage of the particular cells that are demanded by the body. So you're going to find that these hematopoietic stem cells, they will be actually there forming the erythroid. Those erythroids are now that are going to be the committed cell forming now the red blood cells. So erythropoiesis is just a process by which these red blood cells are formed from the hematopoietic stem cells in the bone marrow. So we talked about the structures where they are found. So now let's get to go into the diagram that is going to be showing us, or simplifying some of the terms in erythropoiesis stage. So these are the steps that are involved in erythropoiesis, from this hematopoietic stem cell up to a mature red blood cell. So we're just going to run through all these and look at some of the regulatory factors, and from there we'll be done. So let's start now from the first step. So the first step here, we are looking at this. So basically at this point, we haven't yet entered into the whole process of erythropoiesis, but we are just trying to start from the hematopoietic stem cells to see what happens there, then how it gets now enter into the whole process of erythropoiesis. But it all begins from here. We all have our grandmothers, that's where the whole thing started from. So the first step, or rather the first cell that we are going to look at, is the hematopoietic stem cell. So this cell, at this point, it is not going to have any morphological signs that it is going to become a red blood cell. At that point where you're trying to scan to say, this thing going to become this or that, but it is not actually showing any morphological signs that it is going to become a red blood cell. So there will not be any specific red blood cell features, but due to the presence of certain inducing factors, it is going to differentiate and form this cell, which is known as the common myeloid progenitor cell. And this common myeloid progenitor cell will differentiate to form a committed type of cell, which is the colon forming unit erythroid cell, abbreviated as the CFUE, common unit erythroid cell. So you're going to find that at this stage, the common myeloid progenitor cell can form several types of myeloid cells. These myeloid cells are cells that are capable of differentiating into either red blood cell, platelets, or granulocytes. So it can form it can either form cells that can form white blood cells or platelets, but in this case, it is going to form a cell that will now specifically initiate the whole process of erythropoiesis, which is the colon forming erythroid. And at this point, the colon forming erythroid, it is going to be the first committed type of cell that is only going to be committed to form the red blood cell. And it is going to require a certain molecule, that is a glycoprotein hormone molecule, that is called erythropoietin. So you're going to find that at this step, this cell is going to be having erythropoietin receptor for the binding of that particular hormone. We'll look at it also. So at this point now, the cell is now going to be known to say, definitely this guy is going to become a red blood cell. He's going to graduate and become a doctor. So you're going to find that at this point, this cell is now going to be known to say it is definitely going to become a red blood cell. So from there, stage 1 now will start from the erythropoiesis step. Stage 1, or day 1. At day 1, you're going to have the formation of a cell that is known as a pro-erythroblast. So this pro-erythroblast cell that is going to be formed, it is going to be about 15 to 20 micrometers in terms of its shape, or in terms of its size, rather, not shape, but size. And it is going to be having a basophilic cytoplasm, which means that when this cytoplasm of this particular cell, it is stained, it is going to appear more like blue or purple with basic stains or basic dyes. So when it is put in a specific dye, it is going to reflect a blue or purple color. That is what it is going to, that is what they are saying at this step. This is going to be reflecting a basophilic cytoplasm, and it is going to have a large nucleus, as we have seen. And at this point, there's not going to be any formation of hemoglobin, but the hemoglobin is not yet visible that you can see that it is being formed. But behind the scenes, there's some preparation there. Then from the proerythroblast, then the erythroblast is going to be formed, which is going to be day 2. So day 2, there will be the formation of the erythroblast. This erythroblast is also known as basophilic erythroblast. That is because the cytoplasm is still presenting itself in a basophilic form. Then the nucleus at this point, or rather the nucleoli, which is found inside the nucleus, will start now disappearing. Then there will now be initiation of the process of hemoglobin formation. So hemoglobin synthesis at this point, it will start now occurring. And erythropoietin also has got an effect at this point. So meaning it has got a receptor that tends to act on at this type of, at this stage in the whole process of erythropoiesis. Then from there, we go to day 3. So from to day 3 there, we are going to find that this erythroblast cell, it is going to differentiate into what we call the polychromatophilic erythroblast. This polychromatophilic erythroblast at this stage 3 or day 3, it is going to be approximately about 10 to 15 micrometers in size, and the hemoglobin at this point, it is going to be visible. And then from there, you're going to find that the nucleus of this particular cell will start condensing. It will continue to condense and condense and condense. And from there, you're going to find that it will start now transitioning. So it will start now transitioning to form the structure that will now start looking like a structure of a mature red blood cell, because at this point this guy had just to be ready, just has to be ready to form the mature red blood cell. So its structure start now transitioning towards the structure of that mature red blood cell. So from there, then day 4. At this day 4, we are going to have this cell that is known as a normal blast cell. It is going to be a normal blast cell, and it is going to be about 7 to 10 micrometers in terms of its size. Then you're going to find that at this point, there will be now extrusion of this nucleus that was condensing here. So the nucleus has undergone a process known as pargnosis. So that pargnosis is the condition in which the nucleus shrinks, condenses, and becomes dark, such that now it is going to be extruded out. It is going to be taken out of the cell, because we know that red blood cells, they don't have a nucleus. So this nucleus is going to be taken out, and at this point the cell will start showing or reflecting a size of about 7 to 10 micrometers. Then from there, we are going to have the formation of an immature red blood cell, also known as reticulocyte. At this reticulocyte, the reticulocyte actually takes about 1 to 2 days for it to just mature now so that it can form now a mature red blood cell to go for a war or to go into the circulatory system. So you're going to find that this reticulocyte is going to be about 7 to 7.3 micrometers in terms of size, and it is not going to be having a nucleus. So there will be no nucleus. Why? It is because the nucleus has already been chased out. It has been sacked. We don't need you. So at this point, the reticulocyte is not going to be having a nucleus. And the other thing is that at this point, most of the cellular organelles, or let me just say, there will be lots of cellular organelles at this point. So there will be lots of cellular organelles. So we're going to find a structure that like the mitochondria won't be there, because we don't need oxygen to be used up as it is being transferred to produce energy. We don't want that. Oxygen is needed somewhere else. So mitochondria here is not needed. So you're going to find that structure like mitochondria and other cellular organelles are not going to be present there. And at this point, this guy will now take it into a 1 to 2 days of maturing, so that it can become now the boss, which is the red blood cell. So this red blood cell now it is also going to be called as erythrocyte, as we said earlier, and it is going to be about 7.2 micrometers. So we're going to find that the size of these two is not going to be significantly different, but it is just going to be slightly different. So this one here will be now having matured hemoglobin. Its shape is now going to be in place, like it is going to be biconcave. It is going to be having no nucleus, and it is going to be having a flexible membrane as well. So we're going to find at this point, this guy is now ready to go out there and explore the world. So that's how the whole process of erythropoiesis is going to be taking place to form now the red blood cell. So upon knowing that, there are certain changes that we need to be aware of. As we have seen, the whole process as it is coming down here, it's more like the cell is just shrinking, more like reducing in size, of course. So we're going to find that there will be 1, decrease in size. That's one of the changes. Then 2, the hemoglobin is going to be formed. Hemoglobinization will occur, in which the hemoglobin you are going to see it is going to be formed. The shape of the cell is going to change to become biconcave, so that it can support its function. Then the nucleus will disappear. The mitochondria, the RNA, all those structures, all cellular organelles are going to disappear, so that they can create a great surface area for the transportation of the required contents. Then you're also going to find that the appearance here, this side, we are saying that the cytoplasm will be appearing, to be in its basophilic form, but at this point you're going to find that it will be in eosinophilic form. So eosinophilic form is a condition where the cytoplasm will start now appearing like pinkish, reddish, like when it is put in an acidic dye like eosin. So eosin is one of the acidic dyes in which these cells can be put in to view if it is a mature one or not a mature one. So if it tends to reflect a pinkish or reddish type of cytoplasm, just know that this big one has undergone the required process. So from there now, let's go now to the last part, which is the regulation of the whole process. So now, what are some of the factors that tends to regulate this process? So we have got several factors that are involved in the regulation of erythropoiesis. The first factor, it is known as regulation. So the first factor is known as hypoxia. Hypoxia. So hypoxia basically is just a condition in which there is insufficient oxygen supply to a particular body tissue, or the body tissue receives insufficient oxygen to meet its metabolic demands. It's a condition where the body is saying we need more energy here, but there is low oxygen levels that it is receiving. So that condition in which there is insufficient oxygen supply to a particular body tissue, or reduction in the oxygen levels in the body, is known as hypoxia. So hypoxia basically is one of the factors that can regulate the whole process of erythropoiesis. So we know that oxygen is involved in the transportation of, or rather